My name is Elin Lindhagen, and I work as a senior scientific expert of pharmacokinetics at the Swedish National Authority. And thanks you very much for inviting me to this interesting workshop. And I'm sorry for not being there with you in Rome. So the title of my talk is Harmonization of DDI Guidelines among different regulatory agencies, challenges and opportunities. Apart from working with assessment of clinical pharmacology issues in applications to EMA and to Sweden, I'm also an expert in the ICH M12 working group that is working with harmonization of DDI guidelines. So first a disclaimer, this is my personal view on this subject. And I will start talking about the guidelines we have today and about the ICH work with the harmonized guideline. And then I will give you a general introduction on how DDI evaluation in drug development is performed today, to those of you who are not familiar with that. And I will give some examples of some areas where science is emerging fast and that can be considered challenges and opportunities for the harmonization work. That is endogenous biomarkers, modeling approaches like PBPK, and some treatment modalities that are not covered by the ICH M12. So today, every large region has its own drug interaction guideline. And these are examples. FDA has one, PMDA in Japan has one, EMA has got another one. And over the years, many areas of these guidelines have already been harmonized, but differences still exist. ICH stands for the International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human Use, and M12 is a multidisciplinary guideline for DDI studies. The work aims at harmonization and at an update of requirements in line with recently published science and regulatory efforts. The M12 working group is led by Dr. Madabushi from the FDA and Dr. Ishiguro from PMDA. The expert group consists of members from many regulatory authorities, including 2 representatives from the European Commission, Dr. Versandvoort from the Netherlands, and me. There were also members from the large industry organizations, including EFPIA and Pharm. A draft guideline can be found on the ICH website as well as on the EMA website, and we are now working on an update that will be discussed in an internal consultation at the end of this year. And a new guideline will hopefully be implemented next year. ICH M12 is focusing on pharmacokinetic interactions caused by enzyme and transporter inhibition and induction. Thus, pharmacokinetic interactions or interactions due to pharmacodynamic interactions or interactions due to change of pH or food effect are not covered by the guideline. Focus is also limited mainly to small molecules and thus gives limited guidance for larger molecules. All basic principles are the same as in our current European guideline. The harmonization covers, for example, in vitro requirements, data interpretation like safety factors for interpretation of in vitro data, and some emerging fields. All updates are evidence-based and a lot of scientific references are given. So this is the table of content in the new guideline. First, there's an introduction, and then there's a section on in vitro evaluation, metabolism-mediated interactions, transporter-mediated interactions, and a section on metabolites. And then it's a clinical evaluation section about type of study, study planning, and considerations for that, and some other topics, including pharmacogenetics and special considerations for therapeutic proteins. And then the guideline is discussing reporting and interpretation of clinical study results and the risk assessment and management. And there are also some appendices including detailed recommendations on the in vitro methodologies, and as well as some guidance on predictive modeling and a list of drugs that can be used in vitro and clinical studies. And a lot of references are also given. Now I will show a few slides describing the basic concepts of drug interaction investigations for those of you who are less familiar with it. First, the terminology aspect. In a drug interaction, there is often one drug A, which, for example, is an enzyme inhibitor that influences drug B that is metabolized by that enzyme. Drug A is often called the perpetrator, and drug B is the victim of this interaction. One of the over 1,000 comments that came in the public consultation was that the words perpetrator and victim could be perceived as offensive and that other terminology should be used. So we are discussing to change the words to percipient and object, which I think is already commonly used in clinical applications. So when a new drug is developed, both the victim and the perpetrator aspect has to be considered. When it comes to victim, the most important is to identify which are the major elimination pathways of the new drug, because these are the routes that can be inhibited or induced. This is typically involving a mass balance study with a radio-labeled drug to find the elimination routes, and in vitro and clinical studies to find the responsible enzymes and transport proteins. Clinical DDI studies are typically performed with index perpetrators that are strong inhibitors or inducers that are specific for a particular enzyme or transporter. The effect of other perpetrators is often extrapolated from the index study. Which recommendations that are needed in the SMPC of course depends on the therapeutic index of the new drug. How concerned are we about overexposure or underexposure? Sometimes dedicated interaction studies are also performed with drugs that will be commonly used together with a new product. This is an example from the European labeling for the antiviral drug maribavir. So this is a part of section 4.5, which describes the interaction risks, and it starts out with the victim aspect, the effect of other medicinal products on maribavir. So here the main metabolizing enzyme turned out to be CYP3A4, which was also confirmed in a DDI study with the strong inhibitor ketoconazole, an index study. Even though an increased exposure was observed, it was concluded that the tolerability of higher doses of this drug was good, so no dose adjustment was needed. This conclusion was also extrapolated to other inhibitors. On the other hand, the index study with an inducer, rifampicin, proved a large decrease in exposure. In this case, there was a concern about decreased drug efficacy at lower exposure, and it was concluded that all strong inducers need to be avoided, and that a dose increase is needed with moderate inducers. So now to the perpetrator aspect, and this has a more standardized approach for all drugs. A screening is performed regarding in vitro inhibitory effect on enzymes and transporters, as well as induction of selected enzymes. Based on this, you can sometimes exclude an interaction risk, but if the IC50 of an in vitro inhibition or induction is lower than the clinical plasma concentrations with a certain safety factor, there is a concern that the applicants need to follow up. And sometimes this is done with modeling approaches, and sometimes a clinical interaction study is needed. And this is another part of the corresponding perpetrator text for maribavir. A large screening was performed, and most enzymes and transporters were not inhibited or induced. However, in vitro inhibition of the transporters PGP and BCRP was seen. A DDI study was performed with the PGP substrate digoxin, which increased plasma digoxin levels, and a recommendation to use caution with PGP substrates. And the potential interaction with BCRP substrates was, however, never studied clinically, but a recommendation was made to monitor patients taking sensitive BCRP substrates for an increased risk of adverse events. So now to the challenges and opportunities. Some of the harmonization work has been rather straightforward, as there is a lot of knowledge available in published literature and a lot of experience in the companies. Some areas have, however, been more difficult since science is evolving fast, as well as regulatory policies. I will mention three of them: endogenous biomarkers, physiology-based pharmacokinetic modeling, and the DDI risk of larger molecules. So the first area is the use of endogenous biomarkers instead of dedicated DDI studies when it comes to transporter inhibition. Some endogenous compounds in plasma or urine have been proposed to be substrates for drug transporters and can be measured before and after administration of the investigational drug to provide indication of DDI potential of a drug via this specific pathway. Examples of interesting endogenous compounds are given here, and the one most commonly used by companies appears to be coproporphyrin, CP1, as a substrate for OATP transporters. The work in the scientific community is ongoing to validate and characterize proposed markers in terms of their performance characteristics. This is an example of where CP1 has been used to understand a complex drug interaction. It's a published study where the kinase inhibitor fenebrutinib was found to be an inhibitor both of OATP and BCRP in vitro. A DDI study was performed with rosuvastatin with substantially increased IUC. Rosuvastatin is, however, a substrate of both OATP and BCRP, so it was difficult to know which mechanism that was responsible for the observed interaction. Luckily, CP1 was also measured before and after fenebrutinib administration, and no effect was observed on the levels of CP1. It was thus concluded that OATP inhibition was unlikely to be involved, so the interaction was attributed to BCRP inhibition. The next area under development that I want to mention is physiology-based PK modeling, PBPK. It's a type of model that wants to describe aspects of human physiology, for example, metabolism with the abundance of different enzymes, the blood flow to the different organs, the participation coefficient to different tissues, chemical and biological features of different substances. With such a model, a drug interaction sometimes can be predicted based on other in vitro or in vivo data. So if the company wants to use a PBPK model in a regulatory application, they have to show that the model is qualified for the intended use. And this is described, in the EMA guideline from 2018. What needs to be shown depends on the regulatory impact of the model. Is it just done to describe or justify something? Or is it used to replace a DDI study which would otherwise be requested? In the latter case, the modeling is considered to be of high regulatory impact, and the regulatory hurdle for its qualification is thus rather high. Since this is an evolving area and the traditions and terminologies between regions is still somewhat different, ICH M12 covers PBPK modeling only briefly in Appendix 3. It mentions some examples of potential applications of PBPK, like to characterize the potential for DDIs, to indicate whether a clinical DDI study is needed, and to support some clinical recommendations in the absence of a clinical DDI study. So this is a summary picture from a former presentation from FDA describing in which DDI areas there is most regulatory acceptance of using PBPK. The most commonly used regulatory use of PBPK may be to predict the effect of a weaker inhibitor when there is a clinical data with strong inhibitors, mostly for CYP3A4. Another application is to exclude the risk of a perpetrator interaction based on in vitro data only. And here are some examples of other areas where PBPK has been proposed in regulatory setting. In most of these areas, models are used by the companies and researchers in drug development, but are still not qualified enough to use instead of clinical data in drug applications. For example, PBPK is sometimes used to support quality changes or to predict changes in absorption. PK differences between populations can also be addressed. For example, organ impairment or genetic differences can be incorporated in the models. But as already mentioned, these models are based on many assumptions that may be difficult to verify, and regulatory use has so far been limited, at least in the EU. The ICH-M12 guidance is mostly focused on small chemical molecules, but many of the drugs developed today are larger molecules, such as proteins or oligonucleotides. For these, regulatory requirements are less clear still. When it comes to proteins like monoclonal antibodies, the approach and assays used for small molecules are generally not relevant. DDI risks has to be assessed case by case depending on the pharmacology of the drug. Some protein drugs can influence cytokine levels and thereby affect the expression of CYP enzymes, and this sometimes needs to be studied in a patient population when treatment is initiated. Another type of compounds are antibodies conjugated to small molecule drugs, ADCs. These are briefly mentioned in ICH-M12. These are most common in oncology, but are also developed in the antiviral field. For these drugs, the DDI risk is mostly focused on the small molecule component. For most of these products, the plasma levels of free payload is low, and thus the risk for perpetrator interactions is also low. On the other hand, there may be situations, for example, co-treatment with enzyme inhibitors, where the plasma levels of free payload may be increased and cause safety concerns. Peptides are smaller proteins, often with different chemical modifications. This is a group of compounds with large variability in properties, and they are not explicitly mentioned in any DDI guideline. There is a recent review summarizing DDI risks for recently developed peptides, concluding that the company uses a risk-based approach when investigating the DDI risk for peptides, and that a risk for enzyme and transporter-based interactions is mostly evident for small peptides under 2 kilodalton. The last group of compounds I want to mention is the oligonucleotides. Those are out of scope of ICH-M12 and are not mentioned in the EMA DDI guideline. FDA has, however, published a draft guideline concluding that the DDI risk for these compounds are usually low. They are not substrates for CYP enzymes and usually don't inhibit transporters or enzymes. but the risks have to be considered case by case, depending on the chemical properties and the pharmacology target of the oligonucleotide. So to summarize, ICH-M12 will result in a common regulatory guideline for DDI studies in drug development and will be available during next year. The new guideline will hopefully facilitate drug development and regulatory assessment. Some areas under development are only briefly covered in ICH-M12, and drug developers need to follow science and upcoming regional regulatory communication. This includes, for example, the use of biomarkers instead of clinical DDI studies, the use of modeling instead of clinical DDI studies, and the regulatory requirements for other treatment modalities than small molecules. So, thank you for listening.